Yes, sir.
Yes, sir. Shall I begin?
Yes, sir.
Yes.
Please go ahead, sir.
So the topic of my lecture is virtual screening approach for identifying novel bioactive molecules in drug discovery. So first, we will talk about what is mean by drug discovery. Drug discovery is the process through which potentially new molecules, new medicines are identified. So it involves a wide range of disciplines, including biology, chemistry, and pharmacology. So this is what drug discovery definition according to Nature Drug Discovery Journal. Drug discovery is the process through which potential new medicines are identified. And the drug discovery process involves a wide range of scientific disciplines that includes biology, chemistry, and pharmacology. So now we will talk something about drug discovery strategies followed in the past. So the approaches or strategies followed in the past were like traditional practice, brute force, serendipity, and rational drug design. Traditional practice, there are several drugs from natural product origin were discovered through traditional medicinal practice. And brute force technique is a random screening technique used for discovering sulfonamide is the best example we can say for brute force technique, random screening of large number of compounds. Serendipity, so the example drugs like sildenafil citrate and Alexander Fleming's discovery of penicillin, this we can cite for serendipity, luck of research. Rational drug design, there are several drugs today, even this rational drug design technique is followed mostly. So the examples like cimetidine, omeprazole, proton pump inhibitor, so these drugs we can cite under this. So we will see a few examples of this traditional practice, brute force technique, serendipity, and rational drug design. So before enter into virtual screening approach, the traditional practice, this is the example is atropine alkaloid discovered from Atropa belladonna. Atropa belladonna, this was atropine. This is this alkaloid is obtained from Atropa belladonna plant species. Atropa belladonna, the belladonna indicates it is an Italian origin name. In European cultures, so the people of that time, Europe, they used Atropa belladonna's extract for different purposes. So mainly for the cosmetic purposes, for beautification purpose. So once they apply Atropa belladonna extract, what they get is near the eye area that dilation of pupil occur. So that adds beauty to the woman, dilating the pupil of the eye. And the Atropa belladonna extract intake that also produces, that accumulates a lot of heat, then sweating. So the accumulation of heat and sweating and then hallucination, these are all happens. So the accumulation of heat makes the woman adds beauty. So her cheeks becomes red. So this beautification purpose, this traditional practice, this it gives a lot of information about the pharmacological property of the drug atropine. So whenever the people take atropine, Atropa belladonna extract, they get sweating. So the sweating is decreases. So bodily secretion is decreases because of anticholinergic effect of atropine. So the anticholinergic effect is the decrease in reduction of bodily secretion that is identified from this traditional practice. And the mydriatic effect, that is a dilation of pupil of eye, so that is also discovered. So the hallucination, extrapyramidal effect of the atropine alkaloid. So these pharmacological properties were understood from this traditional medicinal practice or traditional cultural practice. So this is one example. So widening of pupil of eye, mydriatic effect, preventing sweating, accumulation of heat, and then hallucination. Next example for traditional practice is cinchona alkalides. Cinchona alkalides, the quinine, quinidine, cinchonin, cinchonidine, these are antimalarial alkalides. They have antimalarial activity. Cinchona is obtained from Cinchona officinalis, one of the species, right? So the cinchona bark, the bitter tasting bark, so that contains these alkalides, quinine alkaloid, and it has antimalarial effect. So this South Indian Americans, so South American Indians, they discovered this drug. So they understood the antimalarial property or anti-fever property from the observation, their day-to-day life observation. Whenever mountain lions they fell in sick, they used to eat the bark of this cinchona tree and they recover from the illness. So the South American Indians, so they adopted this cinchona bark eating or the preparation of cinchona bark like a decoction. They made tea preparations, and then they, whenever they feel sick, they used to take this cinchona bark and they recover from the illness. So this history or historical point, this literature gives information about the traditional use of cinchona alkaloid for anti-fever property, and this leads to the discovery of quinine alkaloids from cinchona bark. Next, what we can cite is another example is morphine alkaloid. Morphine alkaloid is obtained from Papaver somniferum poppy plant. Poppy plant contains more than 25 alkaloids, and the major alkaloid 10 to 15% content is morphine. Morphine is opioid analgesic, and you all know that morphine has a potent analgesic effect. Apart from this, it can produce euphoric effect, and it also produces the addiction liability. So this is a very controlled substance. So the name of the morphine has come from the god Morpheus. So this is also traditional practice. So in India, you might be observed in temples. Whenever the Indians goes to temple, they offer some herbs to God. So whenever people go to Shiv temple, they take Bel Patra, right? So this are Datura plant, this offered to God Shiva. And whenever people go to Vishnu temple, there they used to offer Tulsi, okay, lotus offer to Lord Brahma. So this is in our Hindu mythology, Hindu traditional practice in temples. So likewise in Greeks, Papaver somniferum poppy plant is offered to the god Morpheus, Hypnos, and Thanatos. Morpheus is a brother of Hypnos and Thanatos, right? Morpheus god. So the morphine alkaloid name has come from the name Morpheus, which is the drug is offered to, the herb is offered to Morpheus, Hypnos, and Thanatos. Morpheus is god of dream, Hypnos is god of sleep, and Thanatos is god of death. So you can understand the pharmacological property of the opium alkaloid morphine. Morphine in low doses, it produces a dream-like effect, that is euphoria, and hypnol, its sleep-producing effect, hypnotic effect is understood from this culture, which is the plant is offered to Hypnos. Thanatos is god of death. So the dose of the morphine increased, it produces the respiratory depressant action that leads to death. So, so from this cultural practice, the pharmacological properties of morphine is understood. So these are the best examples. So the atropine alkaloid from Atropa belladonna, cinchona alkaloid quinine from Cinchona officinalis, and this morphine alkaloid from Papaver somniferum. So these are all the best example. These drugs were identified or discovered through the traditional practice, the literatures, the traditional practices, cultural practice. So Friedrich Adam Wilhelm Setterner isolated morphine crystals from Papaver somniferum plant. So Robert Robinson in 1925 he gave the structure of the morphine structure. So these are some examples. So in such a way that by exploiting the natural products, the drugs were alkaloids were discovered: atropine, quinine alkaloid, and morphine alkaloid. Now the second technique, brute force technique. Brute force technique is nothing but random screening of large number of compounds. The drug identified under this category, one best example is sulfamidochrysoidine. So this is prantosyl rubrum. It is a dye substance. Okay, so this is a prodrug. Prodrug, everybody know what is mean by prodrug. Prodrug is pharmacologically inactive substance. After metabolism, it gets converted into the pharmacologically active compound. So prantosyl rubrum is a prodrug which yields the parent drug or active principle, that is sulfanilamide. So this is discovered by Dr. Gerard Domag. He got Nobel Prize in 1935 in the division of physiology or medicine. So he found this prantosyl to be effective against Streptococcus bacteria, and he treated his own daughter with the drug and then saving her from the amputation of the arm. So this drug was discovered in German IG Farben Pharmaceuticals, and today's this IG Farben Pharmaceuticals incorporated into Bayer Pharmaceuticals. So in those days, German, that industrial revolution that made large number of textile industries where they used the chemical industry, they produced a large number of dye substances. So the dye substances, they screened for different pharmacological effects. So through which they identified prantosyl rubrum dye substance as an antimicrobial agent that leads to a separate category, sulfanilamide, and then from that sulfanilamide, so many sulfur drugs were synthesized and they all become highly active antimicrobial agents used to treat different variety of infections. So initially when sulfanilamide drug was discovered, this prantosyl rubrum, the company was not willing to conduct clinical trial for this drug prantosyl rubrum, but this scientist, Dr. Gerard Domag, he treated his own daughter with this medicine and then come up with the excellent result that's shown to company. And then from there, the prantosyl rubrum drug was forwarded into clinical trial that yielded sulfanilamide. So this is brute force technique, so largely used for randomly screening of large number of compounds, and until 1970s. So this technique is very familiar for identifying novel bioactive molecules, brute force technique, or random screening of large number of compounds. Right, now we'll see the next category is serendipity. Serendipity is nothing but luck of research. Luck of research. So Alexander Fleming is a lucky fellow, got the penicillin drug. He accidentally met with the penicillin drug. So he has no intention of discovering any drug, penicillin. Okay, so he's a microbiologist, so working in his laboratory. From the contamination of his Petri plate, that is Staphylococcus bacterial culture, that is contaminated by another microbe mold, that is Penicillin notatum. Penicillin notatum contaminated the culture plate, and side by side both the Staphylococcus culture and the Penicillin notatum was grown in the same plate. There is a green color exudate come from the Penicillin notatum mold that kills the bacteria of Staphylococcus aureus. So this was identified by Alexander Fleming. Actually, it took a lot of time to understand the observation, what was the fungal species and how the growth inhibition of Staphylococcus aureus was found. So the killing of Staphylococcus culture, he repeated the experiment for many times, but in the same environmental condition when occurs, so he was able to discover that Penicillin notatum. So this is one of the best example for serendipity, luck of research, accidentally met with an excellent drug, penicillin which saves the lives of lot of people during the World War. So Alexander Fleming also received Nobel Prize for this excellent discovery, along with the Florian chain. Another best example for the serendipity is sildenafil citrate. This is slightly different from the case of Alexander Fleming's penicillin discovery. Here, sildenafil citrate drug is discovered with the program of identification of antihypertensive drug. So group leader Simon Kempfel research group was prepared a research project that identification of antihypertensive drug, blood pressure lowering compounds, drugs in the UK Pfizer site, okay, and through this program, so they took the target as PDE3, phosphodiesterase 3 enzyme as a target. And they, after several years of research, they were able to identify one of the potent compound, that is sildenafil citrate. So this sildenafil citrate, when it is pushed into clinical trial, after preclinical trials, it pushed into clinical trials. So in the clinical trial 1 and clinical trial 2, the results were disaster at the time. So even the drug was shown better efficacy in preclinical studies. It was not shown in humans. That phase 1 and phase 2 clinical trials was showing disappointment. But rather in phase 1 clinical trials, the healthy volunteers reported that so the drug is producing side effects when they increase the dose, multiple dose, that is 10 times the dose of the originally used clinical trial dose. 10 times when they increased the dose, the drug was showing multiple side effects in healthy volunteers. So that one of the side effect, that is male erection. So this was something new at the time, and there is no drug found at that time for male erectile dysfunction. So originally the scientists were intended to make BP lowering drugs, antihypertensive drugs, but miraculously they found that remedy for male erectile dysfunction. So for male erectile dysfunction, there is no remedy, no treatment option was found at that time. So before 1998, 2000, right? So this drug, this is something curious. They made the Simon Kempfel research group in the Pfizer. What they made is, then they made clinical trial for this male erectile dysfunction and then carried out clinical trial for this purpose, and they made huge success with this. And the pharmacological activity of this drug, that is male erectile dysfunction by inhibition of PDE5 enzyme rather than PDE3 enzyme. So it is a potent are selective inhibitor of phosphodiesterase E5 enzyme. It doesn't inhibit, or it is more selective towards PDE5 subtype. So hence it increases the blood flow in a particular area. So that leads to male erectile dysfunction treatment. So sildenafil citrate is a blockbuster drug at the time. So this is luck of research serendipity. They were looking for drug for the treatment of BP, blood pressure, right? But unfortunately or fortunately, they got some new remedy for male erectile dysfunction. So this is luck of research serendipity. So so this is one of the techniques, but we cannot depend on luck all the time. That drugs will not come as such. Rational design. Cimetidine and omeprazole are the best examples of rational drug design. Cimetidine is a H2 receptor antagonist. H2 receptor present in the parietal cell, so that is inhibited by the cimetidine drug H2 receptor, that is antagonized by this drug cimetidine. So this was discovered in GlaxoSmithKline. This cimetidine drug, Robin Gennelin research group found this drug, discovered this drug. The approach used for designing this drug is cimetidine is physicochemical property optimization. So the lead compound taken was histamine as such. Histamine is an agonist for this H2 receptor present in the parietal cell, and this is modified, lead optimized with physicochemical properties, and then they incorporated guanidine functional group along with the structure of this histidine, and then they identified cimetidine. So omeprazole is a proton pump inhibitor, and this drug also anti-ulcer drug, and it is more potent than the H2 receptor antagonist cimetidine, because the proton pump is the last step in the acid secretion, which has main role or control over the gastric acid secretion, which is directly inhibited or irreversibly inhibited by omeprazole drug. So this omeprazole drug was discovered in Sweden by Hessel Pharmaceuticals, and today it is Astra Pharmaceuticals. So 1960s, they started drug discovery program in Hessel Pharmaceutical Company, where they developed assays for screening gastrointestinal tract anti-secretory compounds. Assay development in 1970s, originally they started lead identification program. They identified benzimidazole compounds. Initially, they got picoprazole, timoprazole, and then they identified omeprazole, lansoprazole, esomeprazole like drugs as proton pump inhibitor. So the proton pump H plus K plus ATPase, where this proton pump drug, it is a bioprecursor prodrug which makes the sulfur atom of the omeprazole drug bounds to the sulfur atom of the cysteine amino acid residue present in 813th position of the proton pump enzyme. So that makes the sulfur-sulfur covalent bond formation. That leads to irreversible inhibition of the proton pump. So that gives a very effective inhibition of gastric acid secretion. That is another blockbuster drug. So that completely replaced, we can say, that cimetidine H2 receptor antagonist. These are all rational drug design. Rational drug design is most prevalent in the pharmaceutical industries for developing new drugs. Okay, so so far what we have seen is the drug discovery strategies. Drug discovery strategies are like the drugs discovered through what we can say that is traditional medicinal practice, right? brute force technique, random screening of large number of compounds, and then brute force technique. After that, what we have seen is serendipity, luck of research, and rational drug design. Rational drug design is most prevalent. Now we'll see the challenges in drug discovery. Challenges in drug discovery. So before that, we have to look into the picture of this drug discovery pathway. The drug discovery pathway is divided into three parts: hypothesis generation, which includes a target structure identification and validation, and then bioassay development, and then lead compound identification and lead generation. Then the second category, second part is candidate development. So the lead compound is optimized, so the more lead optimization, and then preclinical studies, and then clinical trials, phase 1 clinical trial and phase 2 clinical trial. So this part is drug candidate development. And then third part is commercialization, which includes phase 3 clinical trial, and then submission to drug approval, then global launch, okay, and then global optimization or post-marketing surveillance. So the drug discovery path is divided into three parts. Major part, that is hypothesis generation, candidate development, and commercialization. So this is a long process, and which involves a lot of cost. Cost is the and the time, both are the major factor in our challenges in drug discovery. So US pharmaceutical companies, they spend about 1 billion to bring each of their new drugs to market between 2009 and 2018. So the data studied by some analysts between the year, the data pharmaceutical companies 2009 and 2018. So this was published in Journal of American Medical Association. That is approximate cost to bring a molecule from the laboratory bench to the pharma market. It average cost is 1.3 billion US dollar. Okay, and the team of researchers, they reviewed 63 new drugs and biological agents. They are approved in the US between the year 2009 and 2018. From this, they concluded that the average cost for discovering a new drug, that is approximately, that is 1.3 billion. You can imagine the cost. So the cost for specific treatments means drugs, oncology drugs or immunological drugs, so anti-infective drugs, gastrointestinal tract acting drugs, central nervous system acting drugs, and dermatological acting drugs. So among this, the oncology and the immunological based drugs, so these treatments or remedies, they take a lot of cost. So that is approximately 2,722 billion US dollar. Sorry, million US dollar. Now, the second factor, challenges in drug discovery, is a time factor. So in drug discovery stages, where drug discovery, preclinical trial, and then clinical trial, regulatory approvals. So the drug discovery part, where in the initial preliminary stage, target identification, validation, assay development, lead generation, this takes 3 to 5 years. Preclinical trials, so this includes the pharmacokinetic studies and the pharmacodynamic studies, PKPD, ADMET, absorption, distribution, metabolism, excretion, and toxicity screening. This all takes 1 to 2 years. Clinical trials, phase 1, phase 2, phase 3 clinical trials, this task takes 6 to 7 years. The regulatory approval, that takes 1 to 2 years, and then post-marketing surveillance. So the approximate time duration or average time duration for bringing the drug from the concept to the market, pharma market, it takes 12 years. So the two major factors in drug discovery or challenges in drug discovery. One is cost and the other one is time duration. So now the COVID situation is going on. Okay, so now we require the drugs immediately for the treatment of this deadly disease. But if you consider this a drug development like 12 years, so from this 12 years of time duration, if there is no drug, then the entire population of the world will be wiped out during this 12 years because of this COVID-19 situation. So where we cannot wait for this, we can use the repurposing of existing drugs. This can be done with the virtual screening approach. Virtual screening can make the availability of the drug by better prediction quickly. So the cost of the development of the drug and the time factor both can be effectively reduced by application of virtual screening approaches, the computational approaches. So what is mean by virtual screening? The use of high performance computation to rapidly identify and accurately predict the bioactivity of new drug molecules from large databases or chemical libraries. This is what virtual screening. The application of high performance computation to rapidly identify and accurately predict the pharmacological activity of new drug molecules from large databases or existing chemical libraries. It may be a publicly available chemical library or commercial sources or virtual space. Now we can understand with a simple example, that is what is mean by virtual screening strategy. So you consider ocean filled with livestock, that is fish, okay, fish. So there are wide variety of fishes in ocean, right? So you want to selectively identify a particular variety of fish, that is yellow tuna fish. So for which, for catching the yellow tuna fish, you have to make a specialized net. So for perfect shape and size, so that you can catch only the yellow fin tuna fish. So this is comparable with virtual screening. So the entire chemical space is like ocean. The number of compounds is like a fish. So for the drug of the disease for which you are looking for a selective type of chemicals, okay, so those are like yellow tuna fish, the required one, okay. So for selecting that, you are making a specific net, so that is virtual screening strategy. So that will leads to the success identification of a selective bioactive molecule from the large chemical space that can be aided by virtual screening strategy. So this virtual screening strategy we construct according to our need requirement. So now we'll see what is mean by chemical space. Chemical space, which encompasses all possible small organic molecules, including those are present in the biological system. Chemical space, which includes small organic molecules, even it includes that biological system, small organic molecules present in the nature, that is biological systems, right? The chemical space has grown exponentially with a growth rate of 4.4% annual production rate from 1800 to 2015. So this is not even affected by World War. World War did not stop the growth of addition of new chemical entity to the chemical space. So we have chemical space, that is very huge databases like PubChem database, Chemical database, drug bank, which includes only the drug molecules or drug-like candidates. So this kind of a chemical space you can explore for identification of novel bioactive molecules. So the chemical space, which includes public chemical library, commercial chemical library, approved drugs, and also proprietary drugs. So in these days, with all this technological advancement, the suppliers of chemical libraries, they can offer billions of molecules that can be readily available within a week period for biological screening of against any target. So this can be available. So the approved drugs, FDA approved drugs, nearly 10,000 molecules, 9,335 molecules. This is according to the published data in Drug Discovery Today in 2019. And the public databases like GDB, Genome Database 17, right? PubChem Library, Campbell Database, Chipmunk Database. Okay, these are publicly available databases. Proprietary databases that belongs to different pharmaceutical companies, Eli Lilly, Pfizer, example, Evotec, and AstraZeneca, and Merck. They have massive chemical libraries, and the number of chemical compounds in the library is also shown here. So the proprietary chemical space, commercial chemical space, public chemical space, approved drugs, these chemical spaces can be utilized for screening for against a variety of disease target using your virtual screening for successful development of or discovery of new molecules. So why we need an effective virtual screening strategy? So the discovering identification of a novel bioactive molecule from massive chemical library. This can be compared with this picture illustrated here. So that is identification of a needle from a huge haystack. So how difficult the identification of a needle from the haystack is this is very similar identification of a novel bioactive molecule from massive chemical library. So that is the reason why you require a effective virtual screening strategy for identification of novel bioactive molecule. So now you have the chemical space. Chemical space contains drugs, drug-like molecules, lead-like molecules. This you cannot screen against the biological systems like in vitro, in vivo assays. So it is not at all possible. It will consume a lot of time and lot of money, and nobody will do that. The screening of entire chemical space against so this can be done with the in-silico approach. The first going for virtual screening, construction of effective virtual screening strategy, and then identification of lead molecules, hits. Those hits are screened for pharmacological activity, in vitro, in vivo assay, and then preclinical trials, and then clinical trials. So these steps usually you can follow for reducing the cost of drug discovery and reducing the time duration. Now, selection of virtual screening strategy. So there are virtual screening strategies. Either they are ligand-based virtual screening strategy or structure-based virtual screening strategy. Okay. In drug discovery, we often met with 4 different scenarios. So for some disease, you will have the drug target structure, and ligand, both the information available. So in this case, you can adopt both virtual screening, ligand-based virtual screening, and structure-based virtual screening, are both structure and ligand integrated approaches. So this is possible with the scenario 1, where you have drug target structure and then ligand information, both are available. In the scenario 2, where in this case you know the information of target structure and there is no ligand information is available. So in this case, so it is only possible that you can adopt structure-based virtual screening strategy. In scenario 3, where ligand information is available and there is no target structure information. And in this case, you can use ligand-based virtual screening approach. So in scenario 4, where you don't have information for target and ligand, both the information is not known. In this case, you can use develop homology models and then screening of chemical library against this homology model, and then you can identify molecules for that particular target. So this is in drug discovery, the selection of virtual screening strategy. So the virtual screening strategy, ligand-based virtual screening strategy, structure-based virtual screening strategy are integrating both ligand and virtual screening strategy. So in scenario 1, when you have both the information available, target structure information and ligand information, you can able to use both the methods, okay, ligand based and structure based. In scenario 2, where you know only the target structure information, here you can use only one approach, that is target structure based virtual screening approach. So ligand based, in scenario 3, you use only ligand based virtual screening approach. Scenario 4, where you construct homology models and then screen for identification of novel bioactive molecules. So this is selection of virtual screening strategies. So what are ligand-based virtual screening strategies? Ligand-based virtual screening strategies, more often quantitative structure activity relationship models are used. Safe and safe and molecular electrostatics of molecules, ligand information that is taken into consideration for constructing ligand-based virtual screening approach. So more often that is 3D QSAR approach, that pharmacophore or common pharmacophore hypothesis is used, and then fingerprints, that chem informatics models fingerprints, molecular hologram based approach, ligand based virtual screening. So they can be used in the case of scenario 1 and scenario 2. Okay, and ligand-based virtual screening approach. So the molecular docking is the most frequently attempted method in structure-based virtual screening. So where the scenario 1 and scenario 3 and scenario 4, in 3 cases, all 3 cases, you can use structure-based virtual screening approach, that is docking, scoring, rescoring, or constructing pharmacophores based on the receptor site, target structure site, active site. Typical virtual screening workflow. So this we'll see in the following slides. Now I will show a case study of my own research work, that is virtual screening and identification of selective estrogen receptor modulators. So in this virtual screening, what we did is, so in general, current drug discovery, computational tools are increasingly used. The purpose is cost-effective and rapid identification of new drug candidates. So the development of virtual screening approaches are gaining much attention in these days. So because of their attractiveness in identification of novel bioactive molecules from massive chemical libraries. Virtual screening protocols, they can be designed to meet any specific requirement depending upon the availability of known ligands and target structure information. Unfortunately, there is no virtual screening strategy that exists or that can guarantee high hit rates for all biological targets. But each target that requires a distinct fine-tuned solutions for identification of novel bioactive molecules, constructing a virtual screening strategy. So the aim of our study, brief aim is to compare retrospective manner the effectiveness and usefulness of common pharmacophore hypothesis, molecular docking, negative image-based screening. And the second aim is to test these developed models in prospective discovery of novel selective estrogen receptor modulators or estrogen receptor alpha ligands. Okay, this is our aim. So retrospective manner, we screen the effectiveness and usefulness of the common pharmacophore hypothesis. This is a ligand-based virtual screening approach, and the structure-based virtual screening approaches like molecular docking and negative image-based screening, and application of these models into the prospective drug discovery, that is for identification of selective estrogen receptor modulators and estrogen receptor ligands. So three virtual screening strategies: common pharmacophore hypothesis, this is a ligand-based virtual screening strategy; molecular docking, structure-based virtual screening technique; and other tool is negative image-based screening. This is also structure-based virtual screening approach. So for this purpose, we took a data set. So the data set selected from the compounds were collected from Chemical database, and they are verified with the Merck compound collection. So this set contains 101 molecules. They belongs to different chemotypes. In quantitative structure activity relationship, we require chemical diversity. Chemical diversity is much more important in 3D QSAR. So in pharmacophore model development, and the number of compounds would be higher. Okay, so this is a statistical approach where we use statistics. So if the number of compound is more, then the prediction will be more precision or accurate. And for developing common pharmacophore hypothesis, you not only require the large number of compounds, you do also require the pharmacological activity. The pharmacological activity diversification is also a requirement. So that is, you require a large number of active molecules, large pool of low active molecules, and then some inactive molecules, highly active molecules, low active molecules, and average active molecules. So this kind of pharmacological activity diversity is also required. So in our case, the low active molecule PIC50 value is 5, and the highly active molecules PIC50 value is 9.7. The number of compounds are 101 molecules, belongs to 20 different chemical types. So the Merck compound collection, the data set 1, and for validation purpose, we made another data set. That data set is they're downloaded from Zinc Dude, that is a directory of useful decoys, Dude set for estrogen receptor alpha. So that originally contains 1434 molecules. The active molecules are like 39 active ligands, and decoy molecules are 1395 molecules. Decoys are nothing but dummy molecules. They are structurally very similar, shape-wise very similar, but they don't have the pharmacological activity, that is estrogen receptor alpha antagonizing activity. So now the workflow for construction of common pharmacophore hypothesis. So the 101 ER alpha ligands, they are selected or curated from Chemical database. So they are verified with the Merck literature, originally published literature. And then this data set divided into two parts. One is a training set and test set. The training set, test set, this classification is essential in quantitative structure activity relationship. So the training sets were used for the model development, common pharmacophore hypothesis development, and 30% of the compounds in the test set, they are used for validation of the constructed common pharmacophore hypothesis. The pharmacophore with best predictions were considered and the remaining poor pharmacophores were discarded. So the statistically best pharmacophores used for the validation was done by two methods, internal validation and external validation. Internal validation with the test set method and external validation was performed with the zinc dude set. So this is the pharmacophore obtained. So this is the best pharmacophore. Pharmacophoric features are 5 pharmacophoric features in this model. One acceptor site and 1 positive center, 3 aromatic ring structures. So this AP-RR223 is the best pharmacophore with respect to different statistics. And this another picture, the picture B, that is 4-hydroxytamoxifen mapped into this particular pharmacophoric feature. Selective estrogen receptor common pharmacophore hypothesis. So the statistical parameters R squared value is, that is squared correlation value, that is 0.923. It is near to 1, so it is a best model. With respect to standard deviation, this model is also best, that is standard deviation is very less, 0.317. Fisher significance value, that is 154.7. Then p-value chance correlation, that is 4.47 into 10 to the power minus 44. Okay, so there is a less chance correlation. Q squared internal cross validation parameter, this is 0.822. So this shows the best predictive ability of this common pharmacophore hypothesis. Root mean square standard deviation or standard error, this is 0.431, and R square prediction, external prediction, that is 0.870. So the screening of this dude set, data set 2 with this pharmacophore. So the data set 2 that contains ER alpha active molecules 39 molecules and decoys are 1395 molecules. So screening of this data set against this particular pharmacophore AP-RR223 that resulted recognition of 16 ER alpha ligands, 16 highly active ER alpha ligands, and none of the decoys were recognized by this pharmacophore. This shows the prediction capability of this pharmacophore, AP-RR223 pharmacophore model. But when we relax one of the pharmacophoric feature from this common pharmacophore hypothesis, it recognizes all 16 ER alpha active ligands, but apart from this, this also recognizes 600, more than 600 decoy molecules. So it indicates that all 5 pharmacophoric features are essential requirement for identification of selective estrogen receptor modulators. Okay, so and it also selectively the chemotype, the recognition capability is very selective. It recognizes particular type of molecules, and because the ER alpha active ligands, 39 ligands, among this it recognizes highly active molecules, and some of them are very specific chemotype. So they have structural similarity with this pharmacophoric feature. Next, workflow for molecular docking based virtual screening approach. So the same dataset 1 and dataset 2 used for molecular docking. So the that Campbell dataset ligands were collected from Campbell and zinc duodecite. Okay, so here the target structure that is taken from protein data bank. That is 3ERT is the protein data bank ID of this protein. Okay, so this initially what we did is protein preparation. So the docking was performed with glide software from Schrodinger software program, glide standard precision, extra precision, and then another software used is plant software where chem PLP scoring function. So the docking models are like SP model, XP model, and plants chem PLP scoring function used. So in our screening, what we found is the extra precision method is not producing no better result than the standard precision technique. So the glide XP docking was disposed. We used only standard precision scoring function and plant software's chem PLP scoring function. So both the functions were used for screening the dataset 1 ligands and dataset 2 ligands against this glide SP and plants chem PLP. And the glide SP docking was R squared value was 0.638 was observed. Okay, for this dataset 1 and dataset 2. For dataset, the AUC values are like 0.91. For dataset 2, the AUC area under curve, rock area under curve values are like 0.91 for this glide SP docking. The plants docking also showing similar results, like the R squared value for this is 0.638 screening result, and the plants docking rock AUC values are slightly less than the glide SP. So this shows that glide SP is showing better ligand recognition than plants docking recognition. Okay, so then what we did is we hybridized both glide SP and plants docking. So the purpose of hybridization is to recognize all active ligands. Here in this next picture, here you can see this. So the docking, this graph shows the plot drawn between the glide SP docking score and the activity in the negative scale, PIC 50, right? PIC 50 in the positive scale and the docking score is in negative scale. So the red triangles, they are all dataset 1 ligands, that 101 ligands. PIC 50 values greater than, less than 7.99, and the blue stars that indicates that all active molecules. So the first one is the graph A is plotted for glide SP docking score. The glide SP docking score recognizes all active molecules. Very few active molecules, they were scored low. So by using this glide SP docking score, we are recognizing mostly all active ligands and we are missing few active ligands. In chem PLP docking scoring function, we are recognizing all active ligands and along with a few inactive ligands scored more. Okay, so for this purpose, for the purpose of recognition of all the active ligands, we hybridized both the scoring function, glide SP scoring function, and then chem PLP scoring function. So now in the third picture, graph C that shows the recognition of all active ligands and few inactive ligands. So in in the original screening, what we should not miss any active ligands from chemical library. So that is very important. So that is why this has made this hybridization of both the scoring function glide SP. So disadvantage with the glide SP approach, so that is here we are missing few active ligands. The disadvantage with plants chem PLP, so that is we are recognizing some of the inactive ligands as active ligands, and for recognizing all the active ligands, what we made is we hybridized both the approaches. So here we were able to recognize the pool of all active ligands. So our objective is fulfilled with this molecular docking approach. So this is construction of structure-based virtual screening, most frequently attempted molecular docking approach. Now I'm going to show you the workflow of negative image-based screening. What is mean by that negative image? So, like simply for our understanding that you can consider in olden days, now digital cameras has come. So previously in photograph we get negative, then negative image of our picture, and then that is developed as original picture, right? So that negative image. Likewise, here in visual screening approach, the protein structure, protein's active site, the ligand binding cavity is taken as a negative image by using different computational approaches, and then that negative image is used for screening the large number of chemical libraries. Okay, so this is the concept behind this negative image based screening. So for construction of negative image based screening model, so we used the same protein, 3ERT protein, which is downloaded from protein data bank, and the protein is prepared first by using Ember software program, and then filling the ligand bind cavity with water molecules by using Voido and flood, and then molecular dynamic simulation is carried out for this is a SORT simulation to gather ensemble of 3 ERT protein conformations. So with the optimized water molecules, we took snapshots from the MD, this ensemble of structures, and then this negative images of ligand binding packet is derived using Voido and float, and we added charge with molecular mechanics force field MMF 94, addition of electrostatistics to the shape of these negative images, right? And then at last we have 21 models, and all these 21 models used for optimization by the dataset 1 ligands and dataset 2 ligands. So we disposed some of the poor models based on their recognition or performance evaluation, preliminary evaluation, and the best models, say with the shape and electrostatistics used for final screening of these libraries, and then we ascertain its performance by rock AUC enrichment and the selection of number of heat molecules. So this is shown in the following tabular column. Okay, before that we will see the ligand binding packet. So this is the negative image of 3ERT protein, where this blue color that shows the binding of the shape based cavity, that is 4-hydroxy tamoxifen is bound in this ligand binding cavity. So this shows the binding poses of different program, that is glide SP, glide XP, shape, and then plants. So this will show the performance of our negative image based screening model. So AUC, rock AUC, rock that ROC, that is a reservoir operating curve, area under curve. So this is for a glide SP with the static conformation, protein conformation, this is 0.91. For plants, it shows 0.65. For hybridization, the AUC value is 0.79. So the AUC, rock area under curve, the scale is starts from 0 to 1. So the value which closest to 1 is the better one. So 0.91 is obtained for glide ESP. So the glide ESP has better recognition capability for data set 1 ligands. Similarly, the same value was observed for data set 2, that is a zinc dude set. Okay, so in relaxed protein conformer, this is the first one is static conformation, that is the original conformation, X-ray crystal conformation. The relaxed conformation, that means MD simulation ensemble structures, that is glide ESP, that shows 0.75, and for data set 1, for data set 2, that is 0.89. And for plants it is 6.2, and in both the conformation, static conformation and relaxed conformation, so this shows better, glide ESP program shows better area under curve value. And you can see negative image based screening model. So this shows shape only program, the ligand binding cavity with only shape, so that shows 0.61 AUC. When electrostatistics is added to the negative image based ligand binding cavity, so like addition of 0%, that is 0.58, and then addition of 10% electrostatistics, so now the AUC improves. Okay, so 50%, so that shows 0.97, which is higher than the glide ESP. The recognition capacity for the negative image based model is better than the glide ESP program. So what I'm concluding here with, according to ROC AUC, so the negative image based screening model, that is safe 50% and electrostatistics 50%, that gives AUC value 0.97, better recognition capacity than the docking program. So the major advantage with the negative image based screening is, so this negative image based screening is a fast approach, because this just only matches ligand molecules with the ligand bind cavities shape and electrostatistics complementarity. Just matching shape and electrostatic matching between the ligand binding cavity, that is a negative image based model, and the ligand shape and electrostatic complementarity based approach. And the glide is computationally expensive approach than the negative image based approach. So that is the advantage. So this is a very quick approach, the negative image based screening approach. So from this, I conclude that the negative image based screening is far better than the glide approach. Glide is also better. I cannot say that glide is on, but the better negative image based screening is better because it is a very quick approach. So screening of large number of chemical libraries, which is more efficient in negative image based models than the docking. Docking is computationally expensive. It takes a lot of time. This is all AUC values. Here this AUC and weighting of electrostatistics. Here you can say that 50% that AUC values are like which were near to 1, that is 0.97. ESP weightage 50%, the red color. So that it's yearly recognition. So is also very higher, and it recognizes highly active molecules. So true positives are higher in the case of the negative image based screening. The recognition capacity distinction of active and inactive molecules is better with the negative image based approach. So this is our retrospective validation, retrospective screening. And now I am going to show the prospective screening of ER alpha ligands. So after constructing common pharmacophore hypothesis, molecular docking based method and negative image based method. So the first part is construction of all these methods, and then performance evaluation with the internal dataset and the external dataset, that is DS1 dataset and DS2 dataset. After that, we conclude that common pharmacophore hypothesis, which is a fast approach, okay, but its recognition capacity is very specific. It selectively identifies similar active ligands, okay, the pharmacophore-like structure, and the diverse recognition is less in this case. So that is the disadvantage, and it misses active ligands. In our screening, what we found is it does not recognizes all active ligands, which is essential requirement in screening process. Okay, then molecular docking. The advantage of molecular docking is it identifies active molecules, all active molecules. The disadvantage with the molecular docking is it is computationally expensive. The negative image-based screening, it effectively identifies all active molecules. The better AUC and the early enrichment found for negative image-based screening. The limitation, the advantage with this negative image-based screening is it is very quicker approach. So this can be used for effective screening of large number of compounds, chemical libraries, NIB model. So now we are coming into prospective screening. So we screened two databases. One is our in-house database and the other one is SPECS commercial database. So from the SPECS library, we are not able to find any active molecules. Very few molecules were recognized, and their so docking score also less. Okay, so the screening of millions of compounds, so we were able to find only few compounds from the SPECS library. And we used our in-house library, which contains coumarin molecules, and we predicted the activity of using our docking program and also the negative image-based screening method, where this pictures shows the EC2 synthesized set, that means in-house library, this orange dots, and this SPECS molecules prepared, they are here. Okay, so they are recognized, their docking score like about 9 to 10, okay, and their activity predicted something like 7. So this is our original screening results. So the first compound is from SPECS commercial library. So the glide SP docking score obtained for this is minus 9.39. Plants Chem PLP score is minus 73.80, and hybridized score is minus 7.89. And the PIC50 what we got from our experiment, sorry, the theoretical prediction, this is experimental value. Sorry, this is experimental value. This is 6.6. And for coumarin molecules, 6.1, 5.9, and 6.5. So this is our prospective screening results. By using docking as well as NIB models, we were able to recognize highly active molecules. So the IC50 determination method used in the experimental one and the Merck screening method, both were different. So we cannot compare the PIC50 values numerically, but the active molecules are recognized. Highly active molecules are recognized by GLIDE SP program, PLANSCHEM PLP or hybrid scoring function, and also NIB-based program. So these compounds were synthesized using this reaction scheme, right? Our in-house library of 3 aryl coumarin compounds. So overall, the findings of our work. So that is, here we investigated the effectiveness and usefulness of ligand-based virtual screening strategy and structure-based virtual screening strategy. Ligand-based virtual screening strategy, what we selected was common pharmacophore hypothesis. Structure-based virtual screening was molecular docking, GLIDE SP docking, and chem PLP scoring function, and negative image-based screening model. So the comparison of all these virtual screening methods, we were able to identify ER alpha binders, and we were able to understand the relative advantages and limitation of all 3 methods. So the NIB method was very efficient in prioritizing the active molecules over large number of decoys and also the chemical libraries. So the optimized NIB model that yields better results for early enrichment than the docking models or pharmacophore models. Further, the negative image based screening, which is a typically faster method, therefore the negative image based screening is a feasible option for screening large databases with less computation time and efficiency. So the objectives in drug discovery is to reduce the time duration. So that can be possible with the in silico approaches. Utilization of high performance computation, so that can reduce the time duration. The number of hits screened in pharmacological assays can be effectively reduced, and the best possibility of obtaining active molecules is also higher by using this virtual screening strategies. So with all this information, I conclude my